Welcome viewers. Welcome to the third lecture on prokaryotic translation. In our previous lectures, we have discussed about the various steps of prokaryotic translation machinery. We have talked about the various important components of prokaryotic translation machinery. In today's lecture, we will be discussing about the basic differences in the translation machinery of prokaryotes and eukaryotes. We will talk about the status of the translation machinery under stress. We will be discussing about the various mitochondrial and chloroplast coded proteins. We will talk about the various signal peptides that help in the translocation or localization of the proteins in their proper positions. We will discuss briefly on the various antibiotics that restrict the translation pathway, and finally we will look into the status of a protein. If it is misfolded, then how the system takes care of the misfolded protein. To start with, let's have a look into the differences between the prokaryotic and eukaryotic translation machineries. As we all know that prokaryotic translation machinery comprises of polycistronic mRNA, which means one mRNA codes for more than one proteins, as compared to eukaryotic translation machinery, which is monocistronic, means one mRNA is coding for only one protein. The second foremost difference is coupling of transcription and translation in case of prokaryotes, as because the site of transcription and translation is one and the same. Unlike eukaryotes, where transcription takes place inside the site nucleus and translation takes place in the cytoplasm. So there is no question of coupling between these two processes. Third important difference is in case of prokaryotes, we see that there exists linear polyribosomes, whereas in case of eukaryotes, the polyribosomes are circular in nature. Next difference is in case of prokaryotes, we don't see any capping of the mRNA, whereas in case of eukaryotes, there is a capping at the 5' end of the mRNA. Here in prokaryotes, the start codon is recognized by a sequence called Shine-Dalgarno sequences, means the first AUG codon that lies after the SD sequence is considered as the start codon. Unlike in eukaryotes, the recognition is dictated by the cap site. In case of prokaryotes, the first amino acid is formyl methionine, whereas in eukaryotes, it is again a simple methionine. In case of prokaryotes, the ribosome is 70S, whereas in case of eukaryotes, it is 80S. Next is, if we consider into the elongation factors, in case of prokaryotes, we have got two elongation factors, elongation factor 2 and elongation factor G, whereas here the elongation factors are eukaryotic elongation factor 1, eukaryotic elongation factor 2. And in case of prokaryotes, the shutdown of translation in energy depleted conditions is done by dimerization, whereas in case of eukaryotes, we will see that it is done by restricting the initiation factors. So, after knowing about these basic differences, I would like to highlight on to a special feature which is present within the eukaryotic system, and that is called as internal ribosome sequences. Herein, what we have seen that internal ribosome entry site are the sequences which are present between the 5' UTR and the start codon. And what is the significance or the importance of internal ribosome entry site is that generally the translation machinery is halted under stress, but if under that stress conditions, we really need certain important proteins to be translated, which are not getting translated, it has to be translated, then it is being done taking help of this internal ribosome entry sites. So these internal ribosome entry sites were first discovered in case of certain viruses that infect eukaryotic cells, and we all know that eukaryotic messages are monocistronic, but the viruses were coding polycistronic messages. So people started thinking of that how polycistronic messages are getting translated within an eukaryotic host. Then they could establish the existence of internal ribosome entry sites, and later on it was found that to encode stress related proteins, these sites play a vital role.
Ma'am, we know that protein synthesis is a energy demanding process.
Yeah.
So when the cell is going through energy starvation or scarce energy conditions, then what is the effect on protein translation machinery?
Yeah, rightly said. Energy is in heavy demand during translation process. So system has made very sophisticated mechanisms to shut down translation machinery under stress conditions. In case of prokaryotes, what we have seen that there exist certain basic proteins called ribosome modulating factors. What they do, they bind to the ribosomes and carry on the dimerization of the ribosomes. And as we all know that if they undergo dimerization, then they cannot participate in the translation machinery. In a similar way, if we look into the eukaryotic system, they apply for a different approach wherein the prime focus is on eukaryotic initiation factor 2. As we all know that eukaryotic initiation factor 2 plays a vital role in the translation initiation process, and it utilizes the energy of GTP hydrolysis. And what happens that after it carries on the initiation event, eukaryotic initiation factor 2, which is now tagged with GDP, has to undergo exchange of this GDP with a GTP molecule. And that exchange process is being executed by another factor called eukaryotic initiation factor 2b, and it helps in exchange of GDP with a GTP molecule. But what happens under stress, that there exists a new kinase which comes into play, and it carries on the phosphorylation of eukaryotic initiation factor 2. And if it undergoes phosphorylation, then even if eukaryotic initiation factor B binds to it, it cannot carry on the exchange process. So if there is no exchange of GDP with GTP, then eukaryotic initiation factor 2 gets inactivated, and there is no question of further initiation of translation. So here you can see distinctly the two different ways by which prokaryotes and eukaryotes are taking care of translation machinery, or in other words, I may say they shut down the translation machinery to avoid wastage of energy under stressed conditions. Once the protein synthesis is done, in case of eukaryotes, it becomes important for the protein to justify its localization. If it is a cytoplasmic protein, then it remains there where it has been synthesized. But there are various other organelles like mitochondria, and in case of plants, chloroplasts are also there. So proteins whose target site is mitochondria or chloroplast, or the site other than the site of their synthesis, they have to be translocated to their original site of action where they are going to execute their function. To carry on this process, there need certain signal sequences. These signal sequences actually help in the translocation of the protein onto their original organelles sites. The signal peptides are helping in localizing the proteins onto their target sites. What happens that these signal peptides are being synthesized during the translation process, and when the translation is still going on, these signal peptides which are synthesized first part of translation, they recognizes the signal recognition sequences, and signal peptides tagged with signal recognition sequences, what they do, they reach towards the translocase, and this helps in the translocation of the polypeptide in its linear conformation. And once these proteins are reaching their own sites, then what happens, there exists leader peptidases which chops up this leader sequences, and then the protein gets folded in its native conformation. Mostly the chaperone proteins oversee the protein folding. Means they take care of the protein folding pathway without knowing the perfect 3D conformation of a protein. This is something very interesting, that they don't know what will be the 3D conformation of a protein, but still they are taking care of the folding process of a protein. So they mostly act by two different mechanisms. One class is those that prevent premature folding, and the second class is that attempt to rectify the misfolding. Let's have a look that what are the chaperones that prevent misfolding. this mainly comprises of heat shock protein 70. What it does, it binds to the hydrophobic domains of the proteins and prevents its misfolding. And they release the protein and allow it to fold properly. And the second class of chaperonins, like GRO-E type, what they do, they are taking out the proteins out of the whole crowd. What happens that the proteins are synthesized in huge numbers, and the hydrophobic domains of these proteins start interacting among themselves, leading to misfolding of the protein. So what it does, it takes out the protein of the crowd and it allows it to take care its native conformation. So let's have a look on the mitochondrial transport mechanism. We see that there exists a leader sequence in the N-terminus of the proteins which are targeted towards the mitochondria.
Ma'am, what are leader sequences made up of?
Yeah, leader sequences are mostly comprising of 20 amino acids which are rich in positively charged amino acids. Like lysine or arginine residues are there in every 3 to 4 residues and have no negatively charged residues. The leader forms an alpha helix with positively charged face and a hydrophobic face. So this alpha helix is being recognized by the receptors present onto the mitochondrial surface. So mitochondrial surfaces have got receptors that bind to these leader peptides, and this are helping the transport of the protein through the membranes. Mostly we use two types of translocases called TOM and TIM, where TOM stands for translocase for outer mitochondrial transport and TIM is translocase for inner mitochondrial transport. So after importing the proteins, the leader peptides are chopped up, as I said earlier. Similar events are taking place in case of chloroplasts as well, but there is a nice thing to be told to you that it is only the plant cells that can differentiate between the leader peptides targeted towards mitochondria and leader peptides targeted towards the chloroplast. As experiments have been done, and it has been seen that inside a fungi, if we introduce a gene into the cytoplasm of the fungi which should be targeted towards a chloroplast, but fungi don't have chloroplast, so they are readily getting transported onto the mitochondria. It means the signal peptides between the chloroplasts and mitochondria have got tremendous similarity, but they have got certain differences which can only be recognized by the plant cells. They have got some complex structures, and it has been seen that their structure, their leader peptides sizes are a bit larger. Here also, similar to TIM and TOM protein or translocases which are present in mitochondria, these translocases which are present in chloroplasts are called TIC and TOC, where is translocase for inner chloroplast and translocase for outer chloroplast. None of us are perfect, so ribosomes are also not perfect. They also commit certain mistakes, right? But the error rate of ribosomes is pretty less, as it commits one mistake out of 10,000 readings, right? So if it makes a mistake, what can be the causes of the mistake? One of the important causes of the mistake is that if the two codons have got difference in one of the nucleotides, then there are chances of errors, number one. And number two, the error may be resulting because of frame shifting, or sometimes it goes through reading onto the stop codons. But the question arises that why I am talking so much about mistranslation, as because there are certain weird genes which are getting translated if and only if the ribosome misreads. One such important gene is the Pol gene of retroviruses, and it is expressed only if its prior gene or gal gene is misread, or the translational error occurs, or frame shifting error occurs, and reading through the stop codons are done. Then only we will see the expression of the Pol gene of retroviruses. So this type of mistranslation are creating havoc situations, right? Now we will be talking about another important aspect, as we have been talking every time, that the genetic code is universal and it is conserved throughout. But still there are certain exceptions. So let's have a look on the exceptions in the genetic code. As we all know that UGA, universally it is a stop codon, but we will see that in case of mycoplasma it codes for tryptophan, and in case of euplots it codes for cysteine. On the other hand, we have got UAA or UAG, which are stop codons universally, but in case of paramecium it is coding for glutamine. And CUG, which is universally coding for leucine, in case of Candida it is coding for serine. So there is certain violations of these rules, and this type of exceptions can be seen predominantly in case of mitochondrial genomes. So let's have a look on that. In case of mitochondrial genomes, we will see that UGA, which is a stop codon, in case of mitochondrial genome of fungi it codes for tryptophan, and in case of protozoa, mammals and platforms also it is coding for tryptophan only. And UAA, which is universally stop codon in all cases, but in case of flatworm it is coding for tyrosine. So this type of exceptions are occurring and the list is endless. So nothing can be said to be as fixed. Next we will talk about the various post-translational modifications that are occurring in the system. One such important example is diphthomide, which is derived from histidine, and it is synthesized by post-translational modification. What happens that after the polypeptide chain of eukaryotic elongation factor 2 has been made on the ribosome, one specific histidine residue which is present in eukaryotic elongation factor 2 is getting converted to diphthomide, and it has got an important role to play, as we all know that diphtheria toxin is responsible for restricting protein synthesis, and its target site is only this diphthomide. So this diphthomide is not present in case of prokaryotes. So this is how the system has made it so that diphtheria toxin is infecting us only. As because we have got this post-translational modification and we have created diphthomide, which is the site for ADP ribosylation by the diphtheria toxin, leading to translation restriction. Now I will be coming on to various unusual amino acids which are present within the system. The 21st amino acid, as we all know, the universally we have got 20 amino acids. So the 21st amino acid is called selenocysteine, and it is encoded by UGA. UGA normally reads a stop codon.
So ma'am, how does the translation machinery decide when UGA should be read as a stop codon or as a codon for selenocysteine?
Yeah, the question is very interesting. System obviously should have a system operative in it to understand that where it has to put stop codon or it has to stop translation, or where in place of a stop codon it has to code for a selenocysteine. And that specific component is selenocysteine insertion sequences which are present within the mRNA itself. And what is so special about these selenocysteine insertion sequences, that they are capable of forming stem loop structures, right? And another factor called cell B comes into play, which is capable of interacting with this selenocysteine insertion sequence stem loop structure and the tRNA that codes for selenocysteine. We do have a dedicated tRNA for incorporating selenocysteine, but the thing is to place this charged selenocysteine tRNA onto the UGA, which is supposed to be a stop codon, is a pretty difficult job, and that difficult job is being made easy taking help of selenocysteine insertion sequences and cell B factor. This is what is happening in case of bacteria. But in case of eukaryotes, here also we have got selenocysteine insertion sequences, but they are at a far away distance from the site of incorporation of selenocysteine. Even they are away from the stop codon. But here we will see that there are more factors which are coming together and joining hands together to place selenocysteine onto the stop codon. The 22nd unusual amino acid is called pyrrolysine. It is very newly discovered round about in 2002. It is mostly found in archaebacteria and it is encoded by again a stop codon called UAG. And it was first discovered in the active site of methylamine methyltransferases found in methane producing archaebacteria of the genus Methanosarcina. But as it is being newly discovered and research is still going on, we really don't know the mechanism of its incorporation onto the polypeptide chain, but it is being proved that it has got a separate amino-acyl tRNA synthetase for it. It has got a separate tRNA which is charged with lysine first, then it is being converted into pyrrolysine, wherein various enzymes come together to carry on the conversion of lysine to pyrrolysine. But we don't know the exact mechanism how it is placed onto the stop codons. Now we will talk about various antibiotics in brief and their mode of action in restricting the translation process. So the components like tetracycline and hygromycin B restricts the translation process in case of prokaryotes, and their target site is the 30S ribosomal subunit. Then we have got chloramphenicol, which restricts the peptidyl transferase center of the 50S ribosomal subunit. We have got puromycin that also restricts the peptidyl transferase subunit of the large ribosomal subunit. Erythromycin is another component which restricts the peptide exit tunnel present in the 50S subunit. Fusidic acid is restricting the elongation factor G. Then the list of antibiotics is endless, but why at all we are interested in learning the mode of action of these antibiotics? Because this helps us in establishing the pathway of translation in unknown systems. Now let's have a look onto the degradation of proteins. So long we have discussed about synthesis, translocation and all. As every beginning has got an end, and as every creator generates a pathway of destruction, so is happening inside the cell. That cells that synthesize proteins, they create the destructors of proteins as well, and they are proteases. Their synthesis is not as complex as protein synthesis, but one thing that needs to be done very carefully is the specificity of the process and the pathway in which they are working, that should be carefully controlled. Otherwise, if they commit certain mistakes, then the entire hassle that we have seen is taken by the system to carry on translation will go in vain. So proteases are the enzymes that are degrading the proteins. They are to be synthesized in separate compartments. They should be present in separate compartments from the compartment where translation is going on, or in other words, they should be synthesized as inactive precursors which are activated when they are needed only. So there should be certain taggings into a protein which the system wishes to degrade. So the system should put certain tags onto the proteins which are to be destructed. So generally proteases are found in various locations, like animals secrete proteases into their digestive tracts. These enzymes are synthesized as inactive precursors and they get activated once they are safely outside the cells that are producing them. Example is trypsin. Its precursor is trypsinogen, and second example is pepsin. Its precursor is pepsinogen. So the precursors are inactive. Next example we will see in case of plants that catch insects, or fungi that trap nematodes, and bacteria that live in rotting animal or plant tissues also secrete proteases. These proteases are generally secreted as inactive precursors and get activated only once they are outside the cells of the producer organisms. You all have heard of lysosomes, which are the membrane-bound organelles found inside the eukaryotic cells. They contain a variety of digestive enzymes, including proteases, and their function is in self-defense. When the immune system cells have engulfed a bacteria or a pathogen or virus particles, the vesicles containing these pathogens are merging with the lysosomes, and the enzyme or the degradative enzymes which are present within the lysosomes are responsible for killing of the bacteria or killing of the virus. So, in case of prokaryotes, we will see that proteases which are destined for export are made as precursors and have been cleaved to form active proteases once they are safely outside the cell. Next, proteases in the membrane-bound lysosomes are degrading the ingested material, as I said, but this degradation pathway this is all about the degradation pathway in prokaryotes.
Ma'am, is protein degradation in eukaryotes similar to that in prokaryotes?
The protein degradation pathway in eukaryotes is much more sophisticated. Herein we see the existence of special organelles called proteasomes. Right. These proteasomes make a cylindrical structure. Within this cylindrical structure, actually the proteins which are targeted to be degraded get entry. Now the question arises that the protein that has to be targeted for degradation should have certain tagging on it. So that tagging is done by addition of ubiquitin residues. So all the proteins that are tagged with ubiquitin are destined to get entry inside the proteasome, and inside the proteasome what happens that the proteins gets degraded. This is how we can see that we do have systems to synthesize proteins, as well as we have got a separate, very specific, compartmentalized system to carry on the degradation pathway.